So the first thing was an interesting thing. I don't know if even I've been over this. I wasn't aware of this before, so I thought I would briefly share it. Um, and let me just pull this up. And I know people sometimes complain, why don't I make these things bigger? And I don't think I can with this. Again, this is not my original work. This was from I don't know if it's a website or a Substack or something, and I don't even really know how to access it. I think the person's name, or at least the name they use, is Archit. Not sure how to even pronounce that, but this is clearly his work, not mine. And it's a great way to do things because basically, I don't even know if he's a doctor, a scientist, or what his profession is. It doesn't really matter to me, but he's contacted a lot of the people like virologists who do studies. They often will put their email or their contact information, anyways, somewhere at the end or the beginning of the study. And it's pretty much expected in medical circles that if you have a question about the paper, you can ask them questions about the paper. So this person who goes by the name of Archit, I don't know if that's his name or just something he uses, has taken upon himself to ask virologists questions. So this is the author, or at least one of the authors of this paper, a pneumonia outbreak associated with the new coronavirus of probable bat origin. And as he says, as of February 2024, this pseudoscientific paper has 1.46 million accesses and 13,140 cross references. And here's one of the main authors, a Chinese guy. And you can go and check this email thread on his website or Substack or whatever it is. And again, I don't know exactly how to access it, but I think if you just use Archit, you'll probably be able to find it. So they cannot be overemphasized how important this paper was to the whole COVID narrative and the coronavirus uh, SARS-CoV-2 narrative. This is the so-called index patient. In other words, the first patient who was alleged to have pneumonia from this new coronavirus. And these are the people who, quote, isolated and sequenced this new virus. And so this became the reference virus, reference genome. And the fact that it was isolated came directly from this paper. So there is no more important paper in the whole SARS-CoV-2 COVID narrative than this paper. And so he's communicating with the one of the principal authors of this paper. So first of all, this is an easier way to see it. He asked them for the details of the isolation. Of course, that this was not really an isolation, but he took some unpurified bronchial alveolar lavage fluid, that's stuff from the guy with pneumonia's lungs, and plated it, in other words, inoculated it or put it on this cell line. That's the monkey kidney cell. This is some liver cell line. And there was 24 wells, so 24 different inoculants. And here's the nutrient medium. Here's the fetal bovine serum. Here's the amount of antibiotics and antimycotics. It's 1% and no trypsin. That's the control group. First control group, second control group, same thing. Then he gets to the experimental group. So they didn't inoculate this with anything from the person with pneumonia. So the so-called independent variable, if there is one, is not a virus, because they actually, of course, didn't look for a virus in the snot. It would be the bronchial alveolar lavage fluid. So in the experimental group, where everything is meant to be the same, you see the same 24 wells, same Viros E6 cells, same amount of nutrient broth, same amount of fetal bovine serum. But here they doubled the concentration of antibiotics and antimycotics, at least in the first pass, and then in the subsequent pass, they did back to the original. And they did the same thing here. So we now have two variables. So they had double the amount of antibiotics in the experimental group, antibiotics and antimycotics, amphotericin and gentamicin, I believe, both of which have been shown to cause cytopathic effect. And there was double the amount in the experimental group as in the control, meaning that even the sputum was not the independent variable, because they varied another crucial factor, which actually has been shown to cause the very effect they're looking for. So he asked him about this, and I don't think you can see his response, but you can go and read it. The answer that he gave for why that he used more antibiotics in the experimental group, thereby negating this as a scientific experiment, was that antibiotics don't affect the growth of the cells, which is wrong. And they used the higher concentration of antibiotics and antimycotics in the sample cell, the experimental group, because they had a sample which could possibly have contained bacteria or fungus, which they didn't have in the control group. So there would be no reason to put the increased amount of antibiotics in the control group. That is a ridiculous reason. It may be the reason they used, and it's possible there may have been bacteria or fungus in the experimental group that weren't in the control group, which even then confounds the whole experiment, because how would you know whether the bacteria or the fungus actually caused a cytopathic effect, even if they were dead? And the answer obviously is you wouldn't. And so that's a ridiculous explanation, because obviously that, since we do know, and there are clear papers showing amphotericin and gentamicin cause cytopathic effect, that negates the whole experiment. So this is a fraudulent pseudoscientific experiment. But the thing I didn't know is that he asked him, how many of the wells in the control group and the experimental group actually showed the cytopathic effect. Now, either I didn't know this or I forgot about it. I would have thought that none of the control group experiments wells showed cytopathic effect, and essentially all of them in the experimental group did. So you would see a clear difference. But in fact, according to his response, they used 24 wells for each culture. In the control group, none of them show the cytopathic effect. And in each of the experimental groups, only one well demonstrated CPE per group. Think about that for a minute. 24 different experiments. So out of 48, 24 for the Vero, 24 for the WHO cells. In the control group, no cytopathic effect. In the experimental group, 2 demonstrated CPE, which is basically almost nothing. which means there are so many other possible reasons besides an alleged virus that could have caused one out of 24 of these wells to show some breakdown. They didn't even tell you how they assessed whether it was a cytopathic effect or not. But even if they assessed it properly, you would have to at least consider there may be some other factor, maybe something fell in that well, or maybe they stirred it wrong, or something besides the fact that that somehow proves there's a virus. That is so beyond scientific and so ridiculous. It actually makes me wonder, when you hear things like this, nobody would believe this. Of course, they don't tell you in the paper how many of the wells in the experimental group actually showed CPE. You have to go and ask them yourself. So they don't divulge this information because nobody with any sense would believe this. It actually makes me think, when I talk about the reasons people say things like this, they either don't care about this at all, so they're not interested in the subject, which is obviously not the case, because if you're doing a paper on it, you're obviously interested in the subject and it's your profession. B, you're just incredibly stupid and you can't figure out that changing the conditions in the experiment negate it being a scientific study. And if only one out of 24 show the effect you're looking for, essentially you didn't get the effect and there was no isolation of anything, even on their terms. So you just cannot believe that these people are that stupid to fall for this, which gets into the third category of they have nefarious intentions. And I'm not necessarily saying that's the case, but it's almost to the point where I hope so, because otherwise they are actually that anti-scientific and that stupid, and that is actually hard to believe. So I'm hoping they had nefarious intentions and they carried out what they were told to do. And they, even though they had to actually tell us on some level what they did, it just boggles the mind that they would consider this a scientific experiment, and essentially the whole world bought it, including, as always, the Truth and Freedom crowd, who still use this as the reference genome and say that this paper shows that they somehow isolated a virus in a paper that is really the poster child for anti-science. So hopefully this is very clear, and that's enough of that. And again, you can check it out for yourself.